So I want to first of all thank Travis for going first. I was supposed to do this first. Luckily, he stepped in time. I actually wanted to add on to what I had initially planned to present, which is to add some slides on single-cell RNA-seq. There were some requests, so I've added that, and we'll go through a bit of that towards the end. Ended up having quite a lot of slides, but I was told by Travis that time is not a concern. So I can bore you for 3 hours. Okay, hopefully you won't sleep. Okay, let's get started. Oops. Is it a wrong presentation? No. I wanted to start by telling you a story. Around this time, in actually July in 2011, it's a long time ago, I had the opportunity to go for a month from my Hopkins at the time for a kind of a visit as a visiting researcher to this lovely institute, IFRIC. This is an an institute that's focused on immune research, mostly innate immune system. It's based in Osaka University. And so I got this fellowship to go over there and do research for 1 month. It was a really nice experience. And this was the title of the project that I was given. The keyword here is DNA sensory genes. And these guys were, they really published well, high profile journals. I actually calculated the frequency of their publications, 1 paper every 6 days. Can you believe it? And high profile journals. And the guy who founded TLR4 is the director of this place. Anyway, so I was given this project. They had this data that they had not published yet, which was microarray data. And my job was to use various techniques available for microarray to find the DNA sensory gene. So I did all kinds of stuff and didn't find it. A few years later, somebody found it, published it. So I wanted to go back to the data. How did I miss it? And what did I find? So this was microarray data. That gene was not on the chip. So I would have never found it. And that is the reason why we are today studying RNA-seq, not microarray. All right? You don't want to repeat my mistake. So the big lesson I learned, and passing it over to you, and maybe you were the reason I went there and learned that hard lesson. Anyway, so coming back, I want to give credit to this wonderful resource that I found, and I highly recommend this one to all of you. It goes into real detail. In fact, a lot of the stuff I have here is based from them, so you can easily follow through. They give you the codes, tutorials, really wonderful. Anyway, I'll upload this slide so you'll get this, so don't worry about it. Okay. Now, in order to do this, you will need a bunch of tools, so I thought I might as well list all the tools up front. So these are the tools you'll need, and the URLs where you can get them. Some of them you might be familiar. FastQC, you might be familiar. STAR maybe, SAM tools for sure, I think you've heard about that. IGV, Integrative Genome Browser. And R, of course, everybody should know R by now. And these are the tools or the packages or the libraries that we get from the various resources to do the analysis. Okay. All right, so as usual, it's always good to begin with a definition or a very brief premise to the whole thing. So the original goal of RNA sequencing was to identify which genomic loci are expressed in a cell or a population at a given time over the entire expression range. And this is the key here, without the absolute need to predefine the sequences of interest as it is in the case with cDNA microarrays. And that goes back to the story that I told you. If you were to do microarrays, you need to predefine the genes or the probes. And so if you miss it, you're not going to find it. With RNA-seq, it's different. Okay, now benefits of doing RNA-seq, well, you can quantify, basically you can find differentially expressed genes, compare between conditions. You can detect transcripts that are expressed at a very low level. It's very good for that. And it can potentially, although maybe it's not used widely, to detect and quantify non-protein coding transcripts, splice isoforms, novel transcripts, and even protein RNA interaction sites. Okay. Now, the general workflow to do a differential gene expression analysis, which is something you do at the end, but there's so much that needs to be done before you get to the destination. So this can be largely broken down into two parts. One is the sequencing or the biochemistry part, and the other one is the bioinformatics part. And for the sake of just wholeness, completeness, I will briefly go through these also before we jump onto the bioinformatics part. And those of you who do wet lab, this is an ode to you. Okay, so RNA extraction. Of course, before you can do the analysis, you got to extract your RNA. So before you can sequence, you must extract it and separate it from the cellular environment. And the cellular environment is usually proteins and DNA, so you got to separate it out from there. Now, there are two common methods. The prevalent methods are silica gel-based membranes or liquid-liquid extraction with acidic phenol chloroform. And one key problem with RNA is they're very sensitive or very fragile. I remember during my undergrad days, I worked in a lab, and I was supposed to extract or isolate this RNA. You run the gel, and I was told by my supervisor, you gotta spray on your gloves this RNA, because RNases are everywhere, so you gotta spray to kind of, keep them away. But in this case, it's not just the RNases that you gotta keep away, the DNases also, in case they break down the DNA fragments and it ends up being there. So you gotta be very careful. Working with RNA is never easy, and some people just have the magic fingers. All right, so now once you have isolated your RNA, you want to know how good is your extraction, the quality of your extraction, your preparation. So in the olden days, we would run the gel and we would look at visually the gel to find out whether we did a good job or not. A sign of a good extraction is if you have your 20S RNA, ribosomal RNA and 18S ribosomal RNA, 18S and 20S, I think this is 20S and this is 18S. If you can see them, and this one should be like, the intensity of this should be double of that, then you have a good intact total RNA. And if that intensity starts, going downwards, and you see, you'll end up with a lot of fragments. So if you see something like that with a lot of smaller fragments, that's partially degraded RNA, total RNA. And if you see a lot of them much lower at the smaller scale, mostly at the lower scale, then it's heavily degraded total RNA, right? So that's in the olden days, traditionally. These days, we have a method called RIN, RNA integration number, RNA integrity number, to evaluate how good is our RNA preparation. I think this was something developed by Agilent. They actually have a software to do this calculation. So the RIN number can range from 0 to 10 or 1 to 10, with 10 being the best and 0 or 1 being the worst. Here you can see 10 and you can see 2, which is very close to, so completely degraded total RNA, intact total RNA. So there's a way to determine whether your preparation was good, right? So now, once you're done with that, then you need to prepare your library. So just going back, so you have your cells that you're interested in, your tissue, or that you're interested in, and then you get the cells and you extract your RNA. So you'll have a mixture of ribosomal RNA, mRNA, microRNA, the smaller RNAs there. You've got a bunch of RNA there, and you got to isolate, separate them. So you get the small ones out using some kit, and you use this polyA select ribosome minus kit or preparation that kind of picks up the mRNA. And then once you have your mRNA separated, you fragment them. There are various ways to do that. Once you get them fragmented, you convert to cDNA, and then you construct your library and you send it off for sequencing. And at each of these steps, you have a number of quality assurance measures that you can take to ensure that whatever you're doing is going well. Right. So Now, with regards to that, we must bear in mind that there are different sizes. So there's a very nice kind of a cautionary remark here. The goal of your RNA-seq experiment will determine the most appropriate library preparations. It all depends on what is your goal, right? So make sure you check the most important parameters, including the expected size. Do you know anything about the size that you expect for your RNA? And that's gonna impact whatever you do here. So you want to make sure that you select appropriately. Okay, so prior to sequencing, we will do a lot of enrichment for cDNA fragments that would be appropriate for the Illumina sequencing, and that's mostly 150 to 1,000. But and for a lot of these vast majority of RNA-seq experiments, the RNA is usually smaller than 150. So these guys are going to be strongly underrepresented. So you want to find ways to kind of manage that issue. And it's important, therefore, for you to know what is the size that you're dealing with, so you could do something to enrich these smaller ones. And I think there's some ways to do that. And once you're done, you're settled with that, you're happy, you submit for sequencing. Now, I remember Dr. Nick Virolda the other day, she played a video, so I'm not going to go through, you should still remember. But roughly, it involves 3 steps: library preparation, cluster generation. This is to kind of strengthen the signal, and then eventually you do your sequencing. All right, so here's another cautionary remark. In most cases of differential gene expression analysis, it is important to increase the number of biological replicates than the coverage or the depth. Now, you know that we always talk about depth, 30x. Nowadays, you have 100x. When it comes to RNA, what's more important to have more replicates than be focused on depth. Okay, so that's something I found to be interesting and something that may not be obvious. So do consider that, and you can read those references if you want to know more. Okay, so, but here's some recommendation in case you are wondering what kind of depth would be appropriate. So recommended sequencing depths for typical RNA-seq experiment for different genome sizes for bacteria, 5 single read, 5 times for single read, 10 times for intermediate genome size, and for large ones from 20 to 50. And this is for de novo if you're interested. As a general rule, the more genes with low fold changes that are to be detected, the more replicates are needed. Again, the emphasis on replicates. And as part of this, you should also randomize what you cannot block. Block here means stratification. If you can stratify your samples based on which tissue, compartmentalize them, then it's going to just increase your chances of getting better signals. So block wherever you can, and if you cannot, then randomize, just to increase that spread. Now, once you're done with your sequencing and you have taken care of all of this, the data goes somewhere. You got to deposit the data, or if you have not decided to deposit yet, you can study it and then eventually deposit it. Or if you want to get somebody else's data, there is a database available that provides you with all this data. So the SRA resource sequence read archive, which is kind of mirrored across these 3 different locations, geographical locations, is the place to go if you are interested in these, sequencing reads, and you want to analyze them, or if you want to get hold of other people's data, you can go there. There's an alternative, though, because this is a place for all kinds of, sequencing reads. Even if you are not interested in RNA-seq, you'll also dump it here. So WGS, WES, etc. So this is kind of a primary dump yard for all kinds of sequencing read data. But if you're particularly interested only in expression, there's another database which I highly recommend, the GEO database, Gene Expression Omnibus database, which is available at NCBI. So there you go if you're only interested in expression data. Now, just want to highlight again something that you should know by now. If you get the data from here, if you download the data from here, you're going to get it in a specific format called FASTQ format. All right, not FASTA format. Travis earlier showed you FASTA format. This is FASTQ format, and I think it's inspired by FASTA. So here's how it looks like. So it doesn't look like your FASTA format. This is just a code to kind of pull it out or show the top few lines. But this is the format. So you have this asterisk @ sign followed by the read ID and possibly information about the sequencing run. So then in the next line, you have the sequence basis. And then you have a plus with maybe some additional information. And then on the fourth line, you have quality scores, which are also referred to as Fred scores. Okay, so these are quality scores. Now, the score is called Fred score, or Q, which is proportional to the probability P that a base call is incorrect, where Q has this equation. So a Fred score of 10, what does a Fred score of 10 means? It means it corresponds to one error in every 10 base calls, or 90% accuracy, right? So a Fred score of 20 corresponds to one error in every 100 base calls, or 99% accuracy, right? So the higher the Fred score, the better the accuracy. And how do you find out, like, how do you know what does this translate to? So they're using the ASCII character, so you can do the conversion using this table here. So for the different platforms, there are certain rules you got to follow. I'm not going to go into the detail, but you can read more about this. Okay, so so just a quick recap. You submitted for sequencing. You have your sequencing results. And that's the sequencing result, the image, and then you get the raw reads, the FASTQ. So that's your file FASTQ, and you're going to do some quality checks. So this is actually the overall workflow for differential gene expression analysis. This is the whole workflow. So you start with your sequencing images, then you get your raw reads in FASTQ, and FASTQC is like the quality control that you will do. We'll talk about this later. So you can do quality control at this stage. And then once you're done with that, you're satisfied, you do mapping. You do alignment to a reference genome. All right, so for that, you can use this software called STAR, and you will align the reads to a reference, and the output will be a SAM or BAM format. And you can do additional QC check for the alignment using this software, RCQC and QODS, depending on how you pronounce it. And then once you're satisfied with that, you do some counting. You got to count the frequency of the transcripts, all right, the aligned transcripts. So for counting, you use the software called feature counts, and you will get a read count table, which will be in text format. And you can also do some QC. There are some descriptive plots that you could do to evaluate the quality of this count table. And once you're satisfied with your count, then you do normalization and transformation of your data. And for that, you can use the software called Day6 or EdgeR. And what will you get? You'll get a normalized read count table in this format. And you can do some descriptive plots to evaluate the quality of all that. And if you're satisfied, then you move on to the next step, which is to actually identify the differentially expressed genes. So that's the step that you're interested in finally. All right, so you do DE test and multiple testing correction. We'll go through this later. So again, you can use this software Day6, EdgeR, or Lima, and you'll get a list of fold changes and statistical values, or essentially genes that are differentially expressed. So that's what you've been waiting for. And once you're satisfied with that, this can be in text format. Then you can do a bunch of downstream analysis, a number of which I covered the other day, your pathway analysis, network analysis, ontology analysis that we talked about the other day. So all that can be part of the downstream analysis over here. So there's a whole workflow. Just a quick recap: get your sequencing, raw reads, align the reads, get the count, and normalize the count, then get your list of DEGs, and then you are ready to do downstream analysis. So not too complex if you go through it a few times. Okay. Now here, it's in terms of the quality control. This is a very nice kind of a Q&A stepwise workflow where you can use it to ask certain questions. And if you say yes, then you go this way. If you say no, you go that way. So this is to help you, evaluate whether what you have is of reasonable quality. If not, then what can you do? So these are standard steps that you should go through. These ones are optional depending on what you end up doing, whether you do de novo or whether you do short read alignment. So I'm not going to go through the details of this, but there is already very well established protocol or workflow for you to manage the quality control of your reads, the sequencing reads, whether they are paired end or whether they are single single read N. Okay. Now let's move on. So we talked about, let's see, just quickly go back. We talked about, right, we talked about that. Now let's talk about the alignment part. Okay. So in terms of alignment, there are two options. You can align to the transcriptome, all right? All the transcripts that are available on in the database, we can do that. Now, if you align to the transcriptome, as you know, the transcriptome is exons only, so it will match to whatever exon is available, and that's it. Now, there is actually a problem with this approach. Any thoughts? What could be a limitation of mapping to the transcriptome? Anyone? Why should you prefer this over this? Yes. All right. So I think what you're pointing to is that the limitation would be whatever we have available. So if it is something novel that has, exactly, all right? So things that are not available in the database are going to be missed. So that's almost like the microarray story that I told you, right? So you want to avoid that unless you are really sure that whatever is your research question would be satisfied by that, then that's fine. Otherwise, you go and align to the genome. The only problem with the genome is that you will have these introns to deal with. So you'll end up with things like that. You know, the read 2, because it spreads a little bit over there, it gets cut off, right? But not to worry, because there's software that will help you, manage this. So it's not so much of a big deal. Okay, but this is, of course, preferred over this, unless you have reason to think that you are okay here. Now, I mentioned here with and without alignment. So there are actually two approaches, alignment free and alignment dependent. What I'm talking about is alignment dependent. There are alignment free approaches too. I think Callisto and Salmon are alignment free. So these ones, they use some sort of kind of a word size or k-mers, small words or k-mers to kind of do the job of. So there is some sort of a matching or alignment, but it's not strictly the alignment that we are talking about, not a proper alignment. So you also have those approaches, and here's a comment. While the lightweight mapping tools such as Callisto and Salmon have been shown to perform almost as good as the classic alignment tools on simulated data, it is clear that these programs are still under active development and are prone to spurious alignments, particularly for lowly expressed genes. So use them with care. All right, so now, in order to do alignment, of course, you need a genome, right, to do the alignment, and you need to have some annotations of that genome so your software knows where is it mapping, what is it mapped to, and all that. So, so you can download your reference genome from one of the major databases, and when you download them, there are a few formats that you can get that data and the annotations that accompany the genome. So there are a few formats: GFF2, GFF3, GTF, BED or BED. So this is, of course, an upgrade of that one. And some people say that GTF is like something in the middle of these two. And this is a little different. So you have various formats that you can utilize. I think GFF3 seems to be the more popular one. And this is an example of what the format looks like. So you have like nine attributes and nine parts or 9 components, and the 9th one is broken down to various little parts. So I'm not going to go through that, but it tells you, what is the reference sequence, the source, the type, start position, stop position, score, etc. And if you go to one of these UCSC genome browser, then you could actually, download this, and you see in here, you can actually, define the name, and in this case, it's GFF format, and you get the output. So you can actually download these genomes in those formats from the major genome browsers. So there's a note here, and this was quite interesting for me. Obtaining a correctly formatted GTF file may be one of the most difficult tasks in the entire analysis. Do take this seriously and invest the time to make sure that the GTF file you're using is correctly formatted. Do not take the risk of introducing strange results. All right, so basically, you got to be very careful to, you got to make sure that whatever you're downloading is correctly formatted according to what you require, and that the software is going to be able to understand, this format is able to handle the format. So do take that caution in particular. Okay, now you're ready, you have your genome data, you have your reads, you want to align them. What are the tools you can use? STAR is highly recommended. Unfortunately, it has like more than 100 parameters. You also have an alternative top hat, which is actually a bow tie wrapper. So any of those, there might be others too. And if you are, for example, doing de novo, then I didn't list here, you can use Trinity software. So there are a lot of choices. For the purpose of this lecture, I'm recommending STAR or the document, I mean the paper recommended STAR. So generally the specific choice of alignment tools, this is quite interesting. The choice of alignment tool has relatively little impact on the downstream analysis. So the choice of your GFF file is more important than the choice of the tools. In general, I think they're more or less equal, although there was a paper saying that there could be some differences in some cases. So you may want to go back to that and look into that and see what could be the reasons for those small differences. But in general, it's okay if you use STAR or top hat. Now, once you align, you'll get an output file, as I showed in the workflow earlier, which will be in SAM or BAM format. Now, so SAM stands for Sequence Alignment / Map. Format is a generic nucleotide alignment format that describes alignment of sequencing of reads to a reference. And this is the format. It's broken down into two parts. So you have this header section and you have the alignment section. And the header section has a particular format and the alignment section has a particular format to be followed. So for the header section, these are the rules. I'm not going to go through. You can read this on your own. And for the alignment section, these are the different parts and what they mean. And for the alignment section, for example, the flag. So you have something called the flag, and the flag itself has a bunch of values that you need to be aware of, what it means, and these values are mostly with regards to the type of sequencing, whether it satisfied the various criteria, various requirements for, let's say, pad-end sequencing or single read sequencing. And then there's also another component here called CIGAR, and this gives you detailed information about the alignment itself. So that also has a bunch of kind of format that it uses to present the information, right? So M is for alignment, I is for insertion, but M can also be match or mismatch, I is for insertion, and so on. So you can read this. And this is an example of how they provide, they kind of convert that alignment into a CIGAR string. So A, one match, and then 2 indel, and then 4 match, and then 3 match. So you got 3M over here. And the red ones are insertion, and the blue ones here represent deletion. So the number of optional SAM-BAM fields, their value types, and information stored within them completely depend on the alignment program, and they can vary substantially. So before you do any filtering on any flag, make sure you know how the aligner generated those values. So again, whatever tool you use, please spend some time to familiarize and understand how does it generate all these different values. Okay, so moving on. So we have done the alignment. Now we want to do quality control of the aligned reads, just to make sure that the alignment was done well. So there are numerous ways to do basic checks of the alignment success. And alignment of RNA-seq reads is usually considered to be successful if the mapping is more than 70%. All right? And most aligners will actually give you some sort of a summary report for you to evaluate how good the alignment is. Okay, such as the number of mapped reads. Here's an example of a report from STAR's log file for 96 samples. It gives you a number of input reads, all kinds of stuff. And this line you see in the middle here is the median, the median of those across the samples. And then you should also, besides relying on those reports, you should also visualize the alignment using one of these software just to make sure that it looks good. All right, so you can use the IGV, Integrative Genomics Viewer, genome browser to open up and look at the data, look at the alignment. So it's very important to visually check. So in this case, blue lines indicate insertion and red lines indicate deletion with respect to the reference genome. Okay, so in summary, once you have aligned, you're doing quality control. There are two types of quality control: general, post-alignment, QC. You can actually, combine all of this in a separate software called multi-QC. If you want to see all of them together, there's this software called multi-QC, which does a nice job in kind of pulling all those QCs together. But individually, you can look at the log files produced by STAR, as I showed you earlier, the SAM tool, FlagStat, results from this software that I didn't go through. And you also have some RNA-specific QC, the read distribution using one of these tools, gene body coverage using one of these, and the splice junction using this one. Right, so there are a bunch of different QCs that you got to do to make sure that everything is good before you proceed further. Now, once you're done and you're happy with your alignment, the next step is read quantification, as I mentioned earlier. So we got to count. We got to count the transcripts. Now, to do the counting, there are, of course, software is available. So HT-seq count, which is part of HT-seq, and feature count, they are available for you to use. And HT-seq, for example, offers 3 different modes to evaluate whether it's a match, it's a gene or not. So, for example, in this case, it's gene A. The read is matching to gene A, so it's labeled as gene A. And you have the other mode, gene A, also this gene A. But in some of these modes, the answers are different. What is being recommended is that you use the union mode compared to the others, unless you have reasons to actually go for the other modes. So using these software and the various modes, choice of the mode available, you will be able to do a count of your read with respect to the genes and have a table available for you to proceed further. Okay, so few issues that you will face when you do this count. The main caveats of assigning reads to a transcript: inconsistent annotations of the transcripts, multiple isoforms, that can also be common, antisense or overlapping transcripts of different genes. So there's really no good solution yet. Be careful with your conclusion. If possible, limit your analysis to gene-based approaches. So go for those that are of high confidence. So we are done with the count. Next would be the normalization. So we now need to normalize the data or normalize the counts. So, there are many approaches available. Different tools do it differently. So this is an example, the effect of different approaches to normalize for read count differences. Due to the library sizes, you see various results. So Day6 is what we are recommending here, and you can see how it is actually quite different from some of the others. Here's a note here. While the majority of normalization methods work well, RPMK, reads per kilo million, and total count normalization should be avoided. This is important. In the context of DE analysis, or differentially expressed gene analysis, no matter how often you see them applied in published studies. So RPKM, FPKM, etc. are only needed if expression values need to be compared between different genes within the same sample. Why? Because you actually are correcting for length. And you should only correct for length if you're comparing with different genes. If you're comparing the same gene, the length would be the same. So there's no reason for you to correct for length. Okay? So the per kilo there, that's actually the length, and the other one is kind of the number of counts. All right, so now you have done the normalization. You also need to do some transformation. Right, so transformation, what is transformation? For example, if you have absolute values and you transform them to percentage, that's a transformation. So you need to transform the count in the kind of normalized count to something else. So the popular choice is, of course, log. So due to the relatively, and the reason why we do log is because the expression values can be so wide, the numbers can be so high that you can, it can be very difficult to compare the data. So what log does is it kind of shrinks those numbers to really small scale. So due to the relatively large dynamic range of expression values that you can get from this data, many downstream analysis actually prefer for you to provide log scale following the normalization. Now, which log should you use? Log 10 can be used, but log 2 is more commonly used because they say it's easier to think about doubled values than powers of 10. And the transformation should be done in addition to the normalization. So you don't just normalize and forget to transform, you should do both. Here's an example. So this is untransformed. As you can see, the read values, the expression values can go very high. Big difference there. But when you do a log, very nicely compacted in some small scale numbers. They're much more manageable in the log space. Now, there's also something called regularized log, which is basically still log, but you're doing some sort of a correction with respect to the library size, and it just makes it a little bit more fine-tuned. If you compare this one with this one, it just looks much, much more corrected. And similarly, you should also, here what we're doing is mean SD plot on all samples, just to look at the variation, between the mean and the standard deviation. And if you do the regularized log, it's much more flatter compared to, seeing the hump over there, right? So you should also consider going and using this regularized log, R-log. Okay, now, besides that, you should still, so you're done with the count, let's just recap a little. You're done with the count, you have done normalization, you have done transformation. You should do some visual inspection at this point to make sure that things are still okay, right? So how can you do visual inspection? So an important step before diving into the identification of differentially expressed genes, look just to see whether the basic global patterns are satisfied. So for example, if you have technical and biological replicates, they should cluster closer to each other than, be haywired somewhere else, right? So those with similar experimental conditions should come closer. So there are a few ways to do this: pairwise correlation, hierarchical clustering, or even PCA. So here's some example. This is hierarchical clustering. So you can see the wild types are nicely together and these ones are nicely together. If you ended up getting some mixture, then that tells you that something is wrong and you need to probably go back and see what happened. Similarly here, you have the row counts on a PCA plot, and if you use the R-log, here is also R-log. If you use the R-log values, you can see much nicer, cleaner separation. Although you can still see the separation here, but it's much better with the R-log. And so this seems like, the two groups are separated, so things look okay for you to proceed further. If you get some mixture, then you want to go back. And if you can't really sort it out, you might have to remove a particular sample because you just can't, make the heads or tail of that. Okay, so PCA and clustering should be done on normalized and preferably transform read counts so that the high variability of low read counts does not occlude potentially informative data reads. Okay, so do not use the raw, but rather the normalized data when you want to do this. Okay, now we are really ready for the fun stuff, finally, after so many steps. Finding the DEGs, differentially expressed genes. Now, they say at this stage, there are only two things you're interested in, two things that you really need to do. One, estimate the magnitude, compare the magnitude, find which ones are really different, significant, basically different. And then second step is to ask whether that difference is statistically relevant, statistically significant. All right, so find those that are different, ask if they are statistically significant, and that's it, you're done. All right, so here are the tools that you can use to identify the DEGs, and these are the pros and cons of them. Highly recommended is Day6. Lima, I think, was originally, I'm not sure originally, but it was widely used for microarray, but people have also, I think, used it for RNA-seq, so it does work. But again, there are pros and cons. You can go through this. What's generally recommended is Day6 and EDGE, although they have some differences that you can look and see which one suits your data better. All right, so here's another note. All statistical methods developed for read counts rely on approximation of various kinds, so that assumption must be made about the data properties. For example, majority of the transcriptome is unchanged between the two conditions. So you make that assumption, and if this assumption is not met by the data, then basically things are not going to go well. So there are some basic assumptions that need to be fulfilled. All right, next, going back to the actual magnitude, difference calculation. How do you then calculate and find those that are differentially expressed? So, so we use actually something called regression model. All right, so regression, y equals to mx plus c, the basic linear regression. So using the regression equation, a little more sophisticated equation here. So that's your intercept, that's your gradient, that's your x, and that's your kind of error margin. So you have, this is, let's say, the control. This is the wild type, and this is the experiment, the condition. Right? So you have the values there and the value here. You got to kind of evaluate the two and see if this B1 is significant. So you get an equation created for B0. So B0, so you get the B0, which is, let's say, 6.67, and then plus B1 times X. B1 here for the baseline is set to 0, and for the other one is set to 1. So this will essentially become 0 plus your error margin. So that's essentially B0, and B1 would be whatever is the value plus 1 times X plus E. So there will be some value, and the difference between the two is going to dictate the magnitude of difference. And then you ask whether that's statistically significant. So you calculate these differences for all of them, and then you do the statistics. For the statistics, you make a null hypothesis. There's no systematic difference between the average read count values of the two conditions. And you do t-test. If two population, if you have more than two population, you do ANOVA, and etc. You know that. However, it's highly recommended that you do multiple post hoc tests. What do you call this? Multiple test correction, and Benjamini-Hochberg or Bonferroni, whichever is appropriate. Although Benjamini-Hochberg is the recommended one, you should do that correction in order to get a corrected p-value, because you're doing this test for many, many genes. So it's important that you do correction. Otherwise, your false positive is going to be very high. All right. So note that the input for the DG analysis are the raw read counts. Now, it's interesting because we talked about getting all the way to transformed and normalized, but what you feed into the software is going to be the raw, because the software will do this in the background. And later on, when we talk about the single cell RNA-seq, a lot of it comes already pre-normalized, so you don't really have the raw. So for those, some of these tools are not going to work. You need a different tool that would be able to take the normalized data and then do the rest of the stuff, right? So please load, otherwise you're going to get nonsensical results. Now, once you're done with all that, you know your DEGs, you can do some interesting plots just to see whether it matches with what you're getting as an output. So you can do some frequency of the p-values. As you can see, that the statistically significant ones are here, but there's so many of them. When you do a plot like this, you get to see what are they, and you can see there's so many. Those that in red are the ones statistically significant, and this is called an MA plot, where you kind of do a plot, a log-fold change against the mean expression. And it gives you a nice plot that shows you where those differentially expressed genes are. And they are usually, they have, I mean, by definition, they should be away from that middle line, because that's where the changes are kind of very, very similar or close to each other. These ones, the changes are different, and they can be upregulated or downregulated. And you can also do heat maps if you want to kind of, evaluate further. But this is for the DEGs only. You're only taking the differentially expressed genes, and you want to do some interesting analysis. So A here is, for example, gene sorted according to the adjusted p-value. B here is you add on hierarchical clustering. C here is the same as B, but what you do is you change the values to the z-score, basically the statistical value, and you get something something different, and maybe there's some value for you to analyze that. So if your RNA-seq results are suggesting expression changes that differ dramatically from everything you would have expected, then you should be very cautious. So if these things don't add up, don't make sense, then you got to go back. Maybe you did something wrong, or there's something wrong with the data. All right. Now, here's another thing. Judging your DEG results, if you have results from one software, should you just take that and move on? You could do that, although it's highly recommended that you run this with multiple software and take the consensus of those software. So for example, here, you do see some differences in the results from Lima, DSeq, and EdgeR, and you do see something that's common that intersects all the three. So maybe you want to focus on that one because those are kind of highly likely to be real, significant, whereas the other ones could be real, but they may also be false positives. So you may want to start from there, and that's already a sufficient number to keep you busy. Unless you are done, then maybe you want to come back and look at the others. And these are just different plots where you can identify those, but a Venn diagram should be able to do the job. Okay, so what can you do next? Downstream analysis. So this is just an example. You can do network, you can do Go analysis, you can even do pathway enrichment analysis, which pathways are more important, all right? So there's a whole bunch of stuff that you can do once you have DEG, your DEG list of DEG genes, which we already covered, so I'm not going to go into the detail. Okay, so I'm done with the classical RNA-seq, which is also called bulk RNA-seq. Now we're going to move into single-cell RNA-seq. Are you still with me? Okay. All right, so a bit of a repeat. Transcriptomics is a high-throughput study, we all know that, of RNA molecules that are expressed spatially and temporally. I want to emphasize that because single-cell RNA-seq kind of dives deep into that. All right, so we know all this, so we can skip. What are the advantages of studying transcriptomics? Well, you get to know, you get to study the tissue location, what's going on at the tissue location, the microenvironment, the stimulus, the impact or effect of stimulus at that location, cell development, differentiation, reprogramming, disease conditions. And we also have, advanced technology nowadays, these days, well, not these days, for a while now, to help us dive into all those tissues, cells, and better understand what's going on, much more deeper than what bulk RNA-seq can do. Here's an example. Why would you want to go deeper in terms of cells? Here's an example of an application. For example, cancer diagnosis. You can have a panel of 21 genes, maybe containing 16 cancer-related genes, 5 reference genes. You can use these panels if you know that these genes are good biomarkers, of course, on the risk of recurrence of ER plus breast cancer, whether chemotherapy, radiotherapy be beneficial, and maybe many other cancers can benefit from a panel like that. Here's an example for the Oncotype DX panel. Now, this is just to kind of recap the bulk RNA sequencing. You get your tissue from the body wherever you're interested in studying. You homogenize, you do your sequencing, you do your analysis. We went through this whole thing, so you should have some clarity now. Now, the single cell looks a little different. Sorry. Okay, so the single cell RNA sequencing, you do the same thing. You get your tissue there, but you do some dissociation, and then you do cell sorting, basically to isolate the different cells. Then you also do what do you call this? Labeling them. You label the cells, you sort the cells, and then they are sent for sequencing. Sorry, barcoded. Yes, that's the word. Barcode, and then separate them, and then send them for sequencing. So that's the difference. Now, what are some kind of, here's a schema that explains the differences. In terms of output, you get average gene expression for bulk RNA because you have cells from all kinds of, you have all kinds of different cells in your tissue. You didn't separate them. So your gene expression, differentially expressed genes are going to give you a democratic average of what's going on, not really specific to a particular cell. Whereas for single cell RNA-seq, it's really for a specific cell. So you're really studying what's going on for that particular cell. You can identify cell types and proportion in sample, and the differentially expressed genes are going to be specific to that particular cell. In terms of cost, this is very cheap. This is quite expensive. So you can see not many people are going to be able to do this because of the cost. And because the cost is prohibitive, and also I guess the technology itself may be prohibitive in some way. You can do 100 subjects or more, but you can do approximately like 10 subjects, which is 2,000 cells per subject. And so this is well established, this is complex, you need fresh samples, the bioinformatics pipelines are mature, there's good consensus, this is in rapid development, there are a lot of still challenges out there. And important QC parameters, well, we've gone through this one, you can have issues of empty wells, gene express is low, doublets filtering, gene express is high, dead stress cells, high mitochondrial reads, major cell type identification, some of which I'm not familiar myself. So some of the advantages of studying scRNA-seq, as I mentioned earlier, for cancer. Why cancer? Because it's important in the case of cancer to target the therapy cell by cell, so we can identify determine the composition of the cell. We can find out if there are rare cell types that are maybe masked in general. And you can also find different cancer clones that could be subject to intramural heterogeneity, right? And this has been shown to have clinical significance. Here's an example of what is meant by intratumoral heterogeneity. So you have your oncogenic hit, and then cancer cell clones evolve, and kind of over time, the new ones overtake the old ones. So your therapy doesn't end up being very effective. So one-size-fits-all therapy is what was pursued with this bulk RNA, what we could do with the bulk RNA. So you have an average clone, and your known drug wouldn't be that very effective, so you can have relapse or metastasis. In the case of a single cell, we have an opportunity to do precision medicine, precision targeting, where you have individual clones and biomarkers for each one of those cells. Then you can have drugs that target specifically each one of those. Now, there has been exponential growth of these scRNA-seq studies over the years, and there are quite a number of platforms that have come up. You can see from 2013, the number of publications have gone up. Chromium seems to be the most popular one till 2020. Okay, so now, we need to, one of the things to consider when you do single cell is data integration. Now, why is data integration important? Because you have low number of subjects. Your data comes from low number of subjects, and there's high cost. So through integration, you have an opportunity to increase your data so that you can have some more statistical power there. So increasing number of studies with similar objectives, of course, you focus on those that are related, and journals mandate data deposition. So we have data available that we can take advantage of for this kind of integration. I'm going to give you an example of integration, how it's done. So let's say we have two data sets from Darmanis et al. 2017 and Van Tijker et al. 2017. You can see the cell types that this data set has: astrocyte, immune cell, neuron, oligodendrocyte, OPC vascular. These are the numbers. And we have neoplastic, which is neoplastic is unclear. It can be benign or malignant cancer. Whereas on the other hand, you have normal and just cancer. All right, so it's much broader classification. This is more specific. But we can draw some parallel between the two. Cancer, I mean, this could be, I think these are related, so this could be a subset of that. And these are clearly normal, so they should be subset of that. So we will use one of these integration software, Surat, to do the integration and see what we get. So you apply Surat and submit the data. Okay, so this is before integration. This is how it looks like. It's like a PCA plot. You project it to high dimension, and this is what you get. This is from Darmanis, looking at brain tissue, and these are the breakdown of the different cells. All right, now let's look at the other one. All right, this is Van Tijker's. So you have normal and you have cancer over there, and a bit of that over there. Okay, so this is before integration. Now let's do the integration. So we do see, we are interested in these two, and we want to know whether we could kind of merge those and use them together. Right, so that's the question. So we do integration, and this is what we get. Right, so there is quite a significant overlap that could be utilized and we kind of can merge the data set and increase the size of our data for analysis. So let's break it down. Let's look at the normal one. So when TICA normal is in blue, and then the Darman is normal, but more, with the specific label of immune cell or oligodendrocyte, which we can consider to be normal. So there is a lot of overlap between those. So that's good. We can use them. What about the cancer and the okay. So for the neoplastic and cancer, there's also overlap there. Some of them, they don't overlap, but we do have some nice overlap. So we could also utilize those that overlap. And then you look for those that are overlapping, and you can have 4 types of overlap. You can have a perfect overlap, a partial overlap, adjacent, or no overlap at all. All right. So of course, you would want to focus on the perfect and maybe to some extent the partial. The others would depend on your research question. All right, so then here's an example of the data file that you had, let's say, before integration. So before integration, you would have two data files. So 2 independent data sets. So gene 1 from data set 1, T cell 1, data set 1, T cell 2, data set 1, T cell 3, and so on. And you have like 200 T cells, 100 NK cells, and etc. And here's data set 2, T cells 500. So data cell 2, T cell 1, 2, 3, etc. all the way to 500, here all the way to 200 and so on. So that's how you have your data set. But after integration, you can merge them. But how do you merge them? You have gene 1, data set 1, T cell 1, all the way to 200. And now you have data set 2, T cell 1 here. So it's at the same level now. So they can be utilized together for analysis. They are no longer separate. And that's thanks to these integration tools. So what is the advantage of integration? You, of course, can you have increased statistical power to study differential expression between conditions, and especially beneficial for rare cell type. You probably have small number of them. Another study has some. Another study has some. If you pool them, then you have more, and you can make some conclusions. And of course, it's much more robust. By this, I mean those that kind of don't overlap, you can remove them, so you kind of lose or get rid of those that may be poor signals or noise. And it seems to be a good scientific practice. This is highly encouraged, especially given that it's too costly and we don't have there's not much we can do in terms of growing that data directly through these experiments. So we take advantage of what's available. Challenges in the scRNA-seq integration. We do have many tools, but there are differences in the quality of integration, the runtime performance, lack of user interface, lack of comprehensive, automated, broadly or applicable workflow. So this itself is a challenge. It's not fully matured yet, but of course you can look at the benchmarks and find the best one and use that one for the time being until something better comes along. Here are a list of various single-cell RNA integration tools and with their different features. As you can see, Surat is highly cited, so that should tell you it's probably the most popular or the most, I guess, widely applicable. But that doesn't mean the others are no good. You should look at the others too and see which one is most suitable for the research question that you want to do, because there are some differences in their approaches. And this is available on R. All right, so typical scRNA-seq data analysis workflow is very similar to what I have covered earlier. So I'm not going to go through, but generally you do your pre-processing, and then you do your sequencing. You get your image, then you do your quality control, you normalize, you do some batch correction, you do some feature selection, and then you're down to downstream analysis, and you can have your differential expression analysis too. All right, there is actually, I'm not sure if you're aware of this, NF Core, there's a website or there's a community that actually provides workflows, and NF Core provides workflow for RNA-seq that you can take advantage of. This is for bulk RNA-seq, but they also have for a single-cell RNA-seq workflow. So if you're interested in doing this, this might be a good place to start because they have pool put it in a pipeline that you can automatically run and use. Okay, so and advantages of the NF core workflow: they are modular, scalable, portable, software dependency in containers. They use Docker, Singularity, Conda, so it allows for reproducible research. And of course, there's documentation, code sharing, and they also have a Slack channel. So if you run into problem, you can just say hello and ask questions. Now, where do you get the data for single-cell RNA-seq? You get it from various sources. These are some of the places. So Geo can also provides, this is what I mentioned earlier, but also provides single scRNA-seq data. PangoloDB, single-cell expression atlas from EBI. CancerSea is more for cancer. Cell Atlas Search, single-cell portal from Broad Institute. And of course, some people have private data that they don't share, and you could be one of those. All right, and this is just a snapshot of CancerSea, and the Venn-Tiker data that I showed you earlier comes from here. All right, and that's, I think that's it. So I hope that gave you a quick overview of what is single-cell RNA-seq all about and some ways to navigate through that quagmire. Okay, thank you. Any questions? Overloaded and hungry. Yes.
Okay. So you mentioned that publishers are required to publish their data as well, their raw data, right? So what databases do you usually go to find those data?
So the question was, I mentioned that publishers require, they kind of arm twist you and make you make your data available, publicly available. So for general sequencing data, you can submit them to SRA database. For gene expression, if you have RNA-seq, you can upload there too, or you can also upload to GeoDB. For example, if you have microarray data, you're not going to upload to SRA, you can go to GeoDB and upload there. So there are databases available, they teach you how to do it, they're very, very happy to help you to provide your data there. Okay. Any other question? Yes.
So I know we've done a little bit of RNA-seq analysis in our lab, and the one thing that I've kind of wondered about and that we've ran into is, so you go from zero expression to some expression, no counts to, some counts, or some counts to no counts, and it's infinite change. I know that when you go through the normalization, it turns the zero into non-zero. But can you comment on any of this? Like, how's the best way to handle that you know of these kind of changes?
All right. So we've also done some, and I remember going back a little into the past. We initially used to use log-fold changes, and that's very cursory. It has its own problems. And you also, hear about certain tissues or cells where even very low expression can result in biological activity. So you have these challenges, and log-fold was not good enough. And then I think they're becoming more statistical these days. You have confidence intervals and all that. So we use that, but your question is from zero to some and from some to this. Yes, we do see that. I think we don't really know what's going on unless you have some idea. But what helps is, this consensus across the software. So choosing those that are of high confidence and then just dropping those that we may not be sure what's really going on, especially if you have those that are not expected to jump. You do have some, yeah, you also have these housekeeping genes as controls. So they recommend that, if you have some of these housekeeping genes, you knock them out and you should not see any expression. So they should be really down. So you got to have some positive controls plugged in, some negative controls. So you have an idea whether the results are reasonable, trustworthy. I mean, we can't really make comments for each one of them, but if there's certain indicators, and that's why it's loaded with just so many steps of QC to just increase the chance of making sure that we get some real biological activity that we are picking up. And I think that consensus across the software is one of those that really helps filter away those genes that may not be significant. Okay. Yeah.
Actually, I have a small confusion. So, is this single cell RNA-seq method is applicable for all type of cancer, or just some on more localized type of cancer, say like breast cancer or something like that?
The question is single cell RNA-seq applicable to all types of cancer or some? I'm not going to say I'm an expert on cancer to answer that, but I have a student working on this, and so far, based on our discussions, I think it's applicable in general if you can extract the tissue from that cancer patient and you can then run through the protocol, you separate the cells, barcode them. It should work for any cell. I'm not sure. There might be some restrictions in terms of your cell sorting methods, but I also read that there are so many different cell sorting methods, magnetic, fax, and others. So maybe there are some limitations there that I'm not aware of, but in general, what I read so far, what I've heard is it should be applicable. But I'm not sure for certain. Yeah. Yes.
It just seems a little controversial if you use like multiple different statistical methods and then sort of pool their data together. Like, so if you use Day6 versus if you use HR, which is, I think it tries to not consider outliers or something because it does that mean analysis, versus Day6, which is more, I guess it excludes some part of the outlier analysis. So, I mean, wouldn't it be better to sort of use the same software but on only replicates rather than pooling softwares together?
All right. So the question is, it's kind of controversial to use different softwares that behave differently and kind of get a consensus of these different softwares and then use that consensus result. Yes, those software are different in certain aspects. So I think a way to think about this would be, it's like they have their own restriction, they have their own kind of setup and requirements. And if these certain genes stood to be significant, basically the signal is strong enough to kind of survive the thresholds, the requirements. And you have another one that operates differently and is still stood up, and another one. So it's like, 3 independent evaluations and they all kind of pick the same ones. So it's like saying there must be something there about these genes that all of them are picking the same signal, especially those that overlap. Those that do not overlap, they may be still valid, maybe still reliable. So it's like 3 different assessments. And yes, the approaches are different, but you still come out strong. You still come out to be significant or differentially expressed. Then there must be something about them. And so it's from that perspective. Of course, maybe if you're saying that tools, if they behave the same, you could, I think in general, these tools, yes, they have differences, but some of them, well, if you want, you could focus on those that are much more closer to each other and take those. But also, I think there's also an advantage of taking something that's different and it's like a different way of assessing and you still come out at the top. And the objective of all of them is the same. The approach is different, but the end goal is the same. And maybe as a result, Some of them might be spurious, but if you end up with 2,000, chances are some of those would be relevant. In a nutshell, you're trying to reduce the search space. And of course, if you want to be comprehensive, then maybe that's not the approach. But if you're really looking for some strong candidates from 2,000, the circle, it has reduced the search space, and you go forward, chances are some of them would be significant, and you'll get some idea of what's going on. It's a starting point, and if you want comprehensiveness, then yes, it's not gonna be able to give you all of them. So in terms of sensitivity and specificity, I think this is more focused on specificity, but yeah, sensitivity is gonna suffer. Okay. Any other questions? How are we doing with time? Okay, thank you very much. We can talk.